So this topic is on gene expression. This is one of the more unifying ideas in genetics. We've talked about what a gene is. We've talked about how to take the DNA of a gene and to make a protein. But we haven't talked about this topic, which is the question of whether those pieces of DNA get turned into proteins at any one moment. As we'll see, there's lots of instructions contained within the genome of an organism, and not all of those instructions are being used to make proteins at any one time. So let's get into it and see what we're talking about here. So we can start from a simpler organism like bacteria. That's the main focus of this presentation here. Bacteria are more streamlined organisms than your typical eukaryotic cell. They're smaller, they have smaller genomes. That means less DNA and also fewer genes. So this is E. coli is one of our model organisms for studying bacteria. It's got a lot of history of people researching it. It's got applications to human health, as you might have heard in the news. And this is an organism with a fairly small genome. So we've been looking at humans and other organisms with thousands of genes, and this is in the 5,000, so about a quarter of the genes you'd find in a human. And of those genes, the second question here, you've got thousands of genes that can make thousands of proteins, and any one of those proteins might be needed or not needed at a given time. So it can benefit the organism not to make all the proteins that it's capable of making at any one time. So that's kind of the subject of gene expression, is that not every gene is being turned into a protein at any time. There's some genes that are kept off, basically, and not turned into proteins until they're needed. So of those 5,000 genes in the bacterial E. coli cell, one of the questions is sort of what is really basically required by that cell. So there's some genetic essentials, and there's some genetic luxury built into there. So some of those luxury genes are not strictly required for the cell to be alive, but they can be required at certain circumstances. So this is an experiment that I thought was pretty interesting, where they went through a cycle of making and testing bacterial cells, where they were continually removing genetic information from that cell. And they said, okay, well, is this gene important? Let's take it away and see what we get. And if the cell survived after removing that gene, they would continue and keep trimming down until they got basically to a genome that they couldn't take anything out of genetically, couldn't take any genes away, and still have that cell be alive. Bacteria, as you probably know, are they're haploid, and so every gene in that genome is its only copy of that gene. So we don't have to worry about diploid or dominance and effects like that. Just basically the presence or absence of the gene. They could remove quite a few of these genes. So we started with around 5,000. And as you can see here, they got down to 473 genes. And these are the genes that would be strictly required to keep this bacterial cell alive. And then a big star asterisk footnote on that is that the cell that they're keeping alive is being treated very well. So it has no competition, it has no predators, it has all the food it needs, it has the perfect kinds of food, it has all the vitamins and nutrients it needs. It doesn't have to synthesize amino acids necessarily, it can pick them up from the environment. So this is a cell that can stay alive in a very pampered laboratory environment. Not necessarily a cell that would survive even a day outside of the laboratory, but for the purposes of kind of whittling down to what is the basics of being alive, this is a really good experiment for telling us what are the basics there. So getting down to 473 genes, and as you can see, 531,000 base pairs. So a lot of the bacteria we've seen are in the millions of base pairs for their genomes. This is awful small, so minimal bacterial genome. So of the genes that were whittled away from that bacterial study where they took genes away, we're basically learning that lots of those were not strictly required. And that, again, not required under very pampered circumstances. But there are also a bunch of other genes that are not required under maybe most circumstances, and only once in a while being required. So you can think about one of the examples I use is if you're running a city and you've got your motor pool, all the cars and trucks that belong to the city, they have them year round, but they're only once in a while, certain times of the year, do they call out the snow plows. So they have these snow plows year round, but they don't gas them up. They don't pay someone to drive them. They're essentially not using those snow plows for 9 months out of the year, even though they've got them. That's sort of like a gene that's necessary, but not always. So it's necessary to have in your genome, but not at all times of life. All right, so getting to this illustration here, this is a summary of what it means to do gene expression. So when we say a gene is expressed, that's that split between having the gene in the DNA and turning the gene into a protein. So the process of gene expression, for one way to think about that, you could just kind of strip away all the extras and say that to express a gene means to turn the DNA into protein. And as we saw before in Central Dogma, the DNA is sort of the information instructions, and the protein, once it's made, is the action. So if you stop the DNA from doing its action, that DNA is not being expressed. And another big theme of this process is that cells have some control over whether this happens or not. So we're also going to strip down to really simple terms here and just talk about genes being on or genes being off. So if a gene is on, I'm going to use that as shorthand for the gene is being transcribed and translated, the gene is expressed. And so if the gene is on, that enables the DNA to become the protein, and we'll see some fine details of that process as we go. And if expression is off, or the gene is off, this means that the instructions are still there, but they're not turned into a protein. So in a lot of cases, we'll see some examples where you can basically flip a switch, a molecular switch of on or off for gene expression. All right, so let's say you had these instructions. How do you keep a gene from getting expressed? Or how do you enable a gene to be expressed? So I use the example of the snowplow not being called up in the summertime. Here's another silly example. Let's say you're Batman and you only respond to crimes that are in progress when you get the bat signal. So Batman is always hanging around waiting to do Batman's job, but without the bat signal going on, there's no Batman doing the job. I know, it's a bad example. On a cellular level, if you wanted to make or not make gene expression happen, it comes down to basically enabling or preventing steps along this pathway. So at the top, we've got DNA, and at the bottom, we've got a functional protein. And this is a diagram from your book, and it highlights lots of different ways that the process that goes from DNA to protein, this whole process involved in gene expression, could have lots of steps where it's affected. But realistically, a lot of times we're looking at just a few of these as a lot more commonly encountered ways that expression is regulated. So I've got on here a couple up here, this typical regulation for bacterial genes, big highlight to the step of transcription. So we're going to see lots and lots of examples of transcription being the level where gene expression is controlled. When researchers look and they want to see if a gene is being expressed, they'll often look for evidence of the gene being transcribed. So if you look for RNA being made from a gene, that's often enough of a signal that says, okay, well, the cell has made the RNA, it's going to go on and continue the process until it makes a functioning protein. So this is kind of the biggest one here, I'd say. Transcription, if you don't remember any other parts of the gene expression regulation processes, just remember that transcription part. That's going to be just a super major theme for controlling expression regulation. You might think about that as, there's a logical reason why, is that transcription is an early step in the process. So if a cell wanted to save energy by not expressing a gene, not making the protein, then the best way to save energy is not to make the RNA, and then you would therefore not make the protein. You're saving on amino acids, you're saving on RNA nucleotides, saving a lot of energy by cutting this process off early at the level of where it's already DNA, as opposed to cutting it off right toward the end when it's almost done, but not quite. All right. So we'll kind of come back to this diagram a few times, but remember transcription. So back to the bacterial example, the example of the minimal bacterium. And this is a different way to look at the perspective of what is really essential in a cell. And it turns out that there are some genes that are basically always expressed. And that tells us that they're essential. You really can't turn these off and still be a functioning cell. And they've got a nice term that I think is helpful for thinking about them. They call them housekeeping genes. And a little more formal term for those are constitutive genes. And what this means is that they're really just part of the makeup, the constitution of the cell that has them. And maybe take a second to think about what kinds of genes would need to be on all the time. And so some are highlighted on here. We've got ribosomal proteins, some other ones that we won't have to spell out here, but just the ribosomal proteins might get you thinking about processes that the ribosome is involved in. So it's involved in translation. So an individual gene can be on or off, but at any one time, the cell is transcribing and translating something. So the machinery to make transcription happen, just in the general sense, like RNA polymerase, those genes have to be on all the time. Translation, genes involved in making translation proteins have to be on all the time. So these are the housekeeping genes. They leave the lights on, just like housekeeping or other basic functions. You know, the cleaning still has to happen. Somebody's got to stay and run the power plant to keep the lights on. Really basic functions are these constitutive genes. So these have gene expression that's basically always on and not really subject to regulation in the way that we're going to talk about other ones, these genes. If you're curious, this study shows that they are using these genes to identify bacteria and other organisms living in the environment. So this is a way to, people are looking for evidence of organisms without actually seeing the organisms, but they can look for the DNA that's left in the environment by those organisms. So they did some surveys, and they're surveying the microbial community as to these different groups that are in different colors, shows you different abundances and the various genes they're using to find them, quote unquote, find them. So we never see them, but we see evidence of them in the RNA they left behind. All right, so this is going to be one of our diagrams moving forward, just a general idea of what happens for transcription. Again, we're looking at gene expression that affects whether transcription happens or not. So we're going to think of this as a little bit of a switch, as in an on-off switch. And what's shown here is this kind of peachy enzyme here is the main enzyme involved in transcription. So take a second, make sure you know the answer to that one. Right, that is RNA polymerase. That's the molecule that makes the RNA by transcribing the DNA template. And what's shown is basically if the RNA polymerase is able to attach to the promoter region, which is in red here, then it's able to do its job. So it does transcription, as it says here, and then these little purple lozenges are, those are the proteins that are made by transcription. So basically gene expression enables these proteins to be made, and then they facilitate this biochemical reaction of turning a circle into a diamond. All right, one last thing shown on here is this green molecule, which is enabling RNA polymerase to do its job. So we'll see that as an important part of expression regulation. Oops, there's our labels. All right, so RNA polymerase attaching to the promoter, in this case, enabled to attach to the promoter because that green molecule is there. So it's helping the RNA polymerase to get attached. Okay, so expanding on that illustration, we've got the same situation with a green molecule that enables transcription to happen. And now we've got a name for that molecule. This is called an activator. And I don't want to, there's a whole lot of terminology here. And I want to maybe keep things simple in terms of the terms we're using here, or at least maybe make you not so worried about memorizing them. So we're going to think of a lot of these terms in really black and white, on or off, plus or minus. And one of those kind of on terms is going to be an activator. That's not how you spell that. And the opposite of that is going to be a repressor. And those are two terms for molecules that fall in a much broader category called transcription factors. And we should have seen those before in the topic of transcription. And if you remember transcription factors, their job was to help RNA polymerase get established. So we're seeing just that happen in these diagrams. The activator molecule attaches to a certain piece of DNA that's in this kind of broadly defined promoter region. And because the activator is attached, that allows RNA polymerase to get there and make transcription happen. Now, what also can happen, because this is a regulated transcription, if that activator is prevented from doing its job, so right now the activator is inactivated by this molecule, it's unable, so it changed shape, it's unable to attach to the DNA, and then it's unable to allow RNA polymerase to get on, and therefore transcription does not happen. So we have a situation where transcription may or may not happen, and it's all dependent on, well, I guess a couple of molecules. Number one is the activator. So the activator does its job and transcription happens, or the activator doesn't do its job and transcription does not happen. And the other important molecule here is this diamond molecule, which is actually interacting with the activator, changing its shape, and preventing it from attaching to DNA. So some of the symbols that are going to be used by your book are these molecules like the activator. They have a part of them that's shown at the bottom where they can interact with what I'm going to call the signal molecule. Sometimes it's a little hard to generalize in some of this stuff. So there's a molecule that's really separate from the DNA and other molecules that we see. It's not interacting with the RNA polymerase. It's this other molecule involved in the biochemical reaction that can interact with the activator molecule in this case. And the other part interacts with the DNA. And that would be part of the promoter in the broad sense. And this molecule, this transcription factor, this activator, is able to change shape if it interacts with, so we had that diamond molecule before. If that interacts with the activator, that can cause it to change shape and actually close up or alter the part of it that would normally attach to the DNA. So it's the same molecule, it's going between two states. In the absence of the diamond molecule, it attaches to DNA. In the presence of the diamond molecule, it can't attach to DNA. That's what's being shown in this diagram here. So the top of that is gone for a reason. That's deliberate by the illustrator to show you that it's no longer able to attach to the DNA. All right, so I'd say of the molecules that are most responsible, probably the activator is the most responsible. It responds to the presence of this blue molecule and does or does not attach to DNA, and that affects the whole process. Okay. So when might it be a good idea to turn on or turn off transcription? We'll see some examples, but there are a couple main situations that we're going to kind of simplify this down because it's a complex concept and I don't want to get it too boggy for you. So a couple of processes that are subject to expression regulation. First one is going to be food that could be digested. And so we'll see some cases where cells interact with molecules that they can turn into energy. They don't want to make the enzymes to digest those molecules all the time, but if they encounter those molecules, it's a good time to turn on the expression of the digestion enzymes. Another process is building molecules. And the big point of this one is going to be making sure there is enough. I'm going to say it in a different way, making sure not to run out. All right, so these are going to be our two big examples for processes that are under expression regulation, and we're going to generalize across a lot of genes just thinking of them in these terms. So, right, so if you have a molecule worth digesting, you don't always run into that molecule. Sometimes, as we'll see, there are molecules that can be used for food, but they're not commonly encountered. So one way to think about that digesting of unusual foods is, say you got a fancy silverware set at home, and once in a while you get out the lobster fork. So you have that, but you don't set it out every time. But in the rare cases where you encounter lobster, it helps to have a lobster fork, so I'm told. And you get that one out and then you make use of it. But you don't want to put it out all the time because you'd be putting it out way more times than you ever used it if you just put it out for every meal. All right, so the control of gene expression, another couple of terms here is whether genes can be induced or repressed. And so these are, again, I just want to kind of stick with more of a plus minus thought about these. So plus, to turn something on, gene expression on, is to induce, or induction, or to repress. It's more of a negative or off concept. Biology is full of terms, and I don't think it helps all the time sometimes. All right. We've seen these already, activators and repressors. So the process of turning on transcription, turning off transcription, using an activator molecule, using a repressor molecule, and also these concepts of induction and repression. All right, so we're building up the same diagram we've seen a few times, just building in a little bit more here. Most of this we've seen, so RNA polymerase, the promoter, the gene that's transcription is controlled by the activator is contained over here. This is the gene on the DNA. When transcription and translation happen, it makes the enzyme. The enzyme enables a metabolic reaction. So I'm going to use these terms substrate and product. Those are terms we use for metabolic reactions. So this is a gene that turns on an enzyme that makes a process happen. So it's an enzyme or a catalyst for making circle turn into diamond. And that'll be important also because this process has a little bit of a feedback. So as we saw before, this diamond can go back and interact with the activator. in this case. Another part we didn't talk about before is to the left here. I think it might be bordering on confusing, but I'm going to mention it anyway, that this activator is itself an enzyme, and as an enzyme, there would be a gene for making it. So there's a gene that contains the instructions for making the activator molecule here, and as a result of its transcription and translation, that activator is made. So we'll kind of assume, just for simplicity, we're going to assume that this is always made over here. We're going to look at one level of expression regulation, which is what happens when that activator interacts with the promoter that it attaches to. All right. So with the possibilities, there are basically two ways to have control. One way is to control with an activator, and that's shown on the right. And the other one is to control with a repressor molecule that's shown on the left. Your book uses these red and green colors, red for the repressor, green for the activator. And so this is showing you two ways to have a system that is what we call inducible. So this is, to be inducible means that the gene is usually off. And when I say usually, that's basically in the absence of one of those signal molecules. So we'll get to there in a little bit. In the absence of the signal molecule, there is no transcription, and that could be either because there is a repressor preventing RNA polymerase, or because there's an activator that's unable to enable RNA polymerase. So one of the ways I like to think about these is as kind of a cumulative plus and minus comparison. So a repressor that is able to repress is activated or active, and its job is to repress. So it's sort of inherently a minus molecule, and if it's got a plus, it's able to do its job. And then if you were to multiply a positive by a negative, then you get a net negative here, keeps things off. In the situation where you have an activator controlling, if you have an activator that can't do its job, then that would be an inactive minus activator. So it's a molecule that's sort of normally a plus, but now it's off, and that gets us a net negative. If it helps you to think about things this way, great. If you find this more confusing, get this to make sense for yourself however you can. So both of these, we have two situations where you can be actually controlled by a repressor and off, or controlled by an activator and also off. And in both of those cases, transcription can be turned on. That's the definition of inducible, means that they can be turned on. And in both of these cases, they're turned on in the presence of the substrate molecule. In the left-hand side, the substrate inactivates the repressor. So then we've got a negative on a negative, turning off the repressor. And then if you multiply a negative by a negative, you get a positive, right? So negative times negative is positive. Alternatively, you can have the substrate molecule activate the activator. That's a positive on a positive. And the product of two positives is also a positive. So left-hand box is under the control of a repressor, right-hand box is under the control of an activator. And the results are the same. So this basically shows in living organisms, there's not many ways to get the same results, and because of evolution, they're sort of sometimes a little accidental. And so you can have either way, depending on the gene, you could have it under control of an activator or a repressor, get the same effect in different scenarios. All right. So we have inducible gene expression, off in the present, or off in the absence of the signal molecule. And then these are a couple different ones that are on in the absence of a signal molecule. So I've got at the top here, don't worry about these terms. It's already layers upon layers of confusing, I think, and to add an extra layer of terms to these, I prefer not to go into that level of detail for you. But we still have two cases on the left, under the control of a repressor, on the right, under the control of an activator. This is a different scenario where transcription is usually on. So in the absence of a signal molecule, transcription is on. And we have over here an inactive repressor. So a negative on a negative means that things are continuing, transcription is on. Up here, we've got a, let's see, a positive on a positive, an active activator. Down here, the presence of this molecule, which is now the substrate molecule, activates the repressor. So that's a plus on the minus molecule, gets us a minus. And over here, we are inactivating the activator, a minus on a plus, and that's a net negative. So same results, we've got an on and an off switch, depending on the format of that activator or repressor molecule. And in this case, under the control of the product of this reaction. So substrate circle to product diamond. In our simplified format, this is going to hopefully make a little more sense. Let me take away my annotations.